Hello 515 Diploma students. In your next lab module, you will be looking at real-time RT-qPCR analysis of gene expression in E. coli. Using a tetracycline-resistant strain of E. coli, the same one you used for the PCR experiment in your lab basics module, you will examine gene expression for the bacterium when it is grown in the presence of tetracycline compared to growth without antibiotic. In this video, I will show you the growth of E. coli in different conditions, that is, with and without tetracycline, and the extraction of RNA from cell samples, which you will do yourselves in your upcoming lab. First, I am going to need to inoculate E. coli cultures. I start with a plate streaked with the tetracycline-resistant strain of E. coli. Using a sterile toothpick, I'm going to pick a single colony of E. coli and use that to inoculate a small starter culture in liquid media. I let the culture grow by incubating it overnight at 37 degrees Celsius with shaking. Once I've grown the starter culture, I will use that to inoculate larger cultures. I've prepared 2 flasks containing 10 milliliters of media each. To one of them, I'm going to add tetracycline to a final concentration of 10 micrograms per milliliter. I'm using a 1% volume of inoculum from the starter culture to inoculate the larger cultures. That is, I'm diluting volumes from the starter culture 1 in 100 into 10 milliliters of media for the larger cultures. Again, I'm letting these cultures grow overnight at 37 degrees with shaking. Now, once the cultures have grown, before harvesting the cells, I'm going to add an RNA preserving solution called RNA Later to the culture in a 2:1 volume. Then I transfer the cultures to Eppendorf tubes and then harvest the cells by centrifugation, making sure to balance the tubes. The cells collect on the bottom of the tube as a pellet, and I can remove the supernatant and discard it by pipetting, leaving only the cell pellet, which I can freeze and save for later RNA extraction. When you are ready to do the RNA extraction, keep the samples on ice. The first thing that you are going to want to do is wash the cells to remove traces of the RNA later preservative. Do this by pipetting 200 microliters of TE buffer over the cell pellets and resuspending them by pipetting up and down. Pellet the cells again by spinning the balanced tubes on a mini centrifuge for 1 minute. Remove the supernatant by pipetting. Next, we need to break open the cells to extract the RNA. We do this by an enzymatic digestion of the cell wall. Lysozyme and proteinase K are enzymes that break down the peptidoglycan of bacterial cell walls. First, we resuspend the cells in each tube in 100 microliters of lysozyme solution. Then we add 20 microliters of proteinase K solution. Incubate the samples for 10 minutes at room temperature, vortexing every 2 minutes. After incubation, add 240 microliters of RNA lysis buffer. And vortex the tubes vigorously. In the following step, we're going to use the yellow spin away filter to clear the lysate, removing most of the cell debris and most of the genomic DNA. Before this step, we'll label the filters. Apply the lysate to the filters placed in collection tubes and then centrifuge them at 10,000 G for 1 minute. After centrifugation, the cell debris and most of the genomic DNA is retained on the filter while the RNA is in the flow-through. This is important. We save the cleared lysate in the flow-through for the next steps of the RNA purification. To the cleared lysate in each tube, we add 1 volume of 95% ethanol and mix well by pipetting. Then, we apply the mixture onto the green Zymo spin column. Now, when you centrifuge these columns, the RNA will stick to the column while other contaminants will pass through. This is important. You want to save the column with your RNA and discard the flow-through. Add 400 microliters of RNA wash buffer to the columns and centrifuge again to get rid of more of the impurities. One of the major impurities in your RNA samples is contaminating DNA. You can get rid of this by enzymatic degradation using DNase 1, which specifically digests DNA. Prepare a DNase 1 reaction mix by mixing 10 microliters of DNase 1 and 150 microliters of DNA digestion buffer in an Eppendorf tube. Apply 80 microliters of the mixture to each of the zymospin columns to which your RNA sample is bound. Incubate the columns at 37 degrees Celsius for 30 minutes to digest the DNA. Then continue washing each column, first by applying 400 microliters of RNA prep buffer and centrifuging, discard the flow-through and keep the column. Then apply 700 microliters of RNA wash buffer to each column and centrifuge. Again, discard the flow-through and keep the column. And for one last wash, apply another 400 microliters of RNA wash buffer to each column and centrifuge. Then take each column and place it into a clean, labeled, and chilled tube. To elute the RNA, add 50 microliters of water that has been treated with DEPC. This will have removed any traces of ribonuclease activity. The RNA will unbind from the column and, when centrifuged, can be collected in the eluted flow-through. This is your purified RNA sample. It can be protected from degradation by adding a ribonuclease inhibitor called RiboGuard. On this sample of RNA, you will perform real-time RT-qPCR analysis. But before you do that, you'll need to measure the quantity and quality of the RNA in subsequent steps that you will do yourself.